All right, folks, in this video, I'm going to talk about transcription in bacteria. So what I'm going to draw for you is essentially a cartoon of cistron, or a protein coding gene, and the resulting messenger RNA, without actually talking about the process that gets us from the DNA to the RNA. That's a subject for another video. But imagine, if you will, that we've got a bacterial chromosome running off in both directions, but for the purposes of this drawing, we know that there is a protein coding gene in this region. So we've got our double-stranded double helix. So how do we know that there is a gene to be transcribed in here, that we want to make that messenger RNA copy? Well, there's going to be a promoter region at about 35 nucleotides ahead of the place where the transcription is actually going to start. So up at the -35 region, we're going to find that promoter. And then there's going to be another important location that's at -10 from the first nucleotide that is going to actually get made into the messenger RNA. So then we'll get to -1, and there is no nucleotide 0. So we go from -1 to +1. Okay, so this region up here is the promoter. And this region at -10 is known as the Pribnow box. Which is analogous to a Tata box in eukaryotes. But this promoter region is the site where DNA strands are going to be separated so that the messenger RNA can be synthesized. It's also going to help orient our RNA polymerase in the proper direction. So we know that this is our transcription unit here, but how do we know that there's not one, that the RNA polymerase is supposed to go this way and not this way? Well, it has to do with the orientation of the promoter in the Pribnow box. Okay, so this is going to be our coding region, which is going to lead to a trailer and then a terminator sequence. So our transcription unit does start at +1, but the initial, the start codon is not going to be at +1. The start codon is going to be a few nucleotides in, because we do need a bit of a lead-in to orient the messenger RNA when it gets to the ribosome. Okay, so this top strand here is called the sense strand, but that's not what we're going to copy to make the messenger RNA. So we know that the start codon is AUG, so here's the DNA version of that. There's ATG. So its reverse complement is going to be CAT, going in the 5' to 3' direction, or TAC is how it's going to get read by RNA polymerase II, okay? So this is our sense strand with DNA versions of codons. But this is going to be, this lower one is going to be our template strand. That's what the RNA polymerase is going to recognize to make the messenger RNA. Which means down here, right before the trailer, we're going to have the DNA version of a stop codon, which one of the ones I picked TAA instead of UAA, and the reverse complement to that is going to be TTA. And then we're going to have a trailer, which is going to be a little bit of downstream untranslated region before we get to the terminator sequence, and I'll talk more about how termination occurs in a separate video. So what this means is that when we get our messenger RNA is going to be single-stranded. It's going to be built 5' to 3'. And it's going to be copied based on what complements our template strand. All right, so our AUG is going to be a few nucleotides into our messenger RNA. And just upstream from that, there is going to be a sequence in the leader, the 5' untranslated region, called the Shine-Delgarno sequence. And the Shine-Delgarno sequence is going to help orient the messenger RNA in the ribosome. The small subunit binds the 16S ribosomal subunit. Sorry, the 16S ribosomal RNA is what binds the Shine-Delgarno sequence and orients it so that the AUG ends up where it needs to be to initiate translation. So we end up with a bit of an untranslated region called the 5' or upstream untranslated region, UTR. Then when we get to the stop codon, that's where translation is going to stop, and there's going to be a 3' untranslated region, or downstream untranslated region. After the stop codon. So the transcription unit does not start with the first start codon and end with the stop codon, but there's going to be a little bit of leader going into the start codon and a little bit of overhang past the stop codon.